Now let us keep a name on that. What name?
I think it should be.
No. Actually, it is all related to nucleus. So why don't we keep it as central dogma?
It's good.
Central dogma.
Okay.
Now let us know what actually central dogma is. It is basically composed of two processes: transcription of DNA into mRNA and translation of mRNA into amino acid and protein synthesis. Starting off with the replication of DNA, the replication of DNA starts at the ORI.
And DNA.
The enzyme nuclease splits the two strands into a DNA, forming a Y-shaped replication form. One of the strands is known as the leading strand and the other is known as the lagging strand. The tension created due to unwinding in the coiled part is reduced by topoisomerase. Thereafter, the DNA primase binds to the helicase, forming the RNA primer. Now the elongation is catalyzed by DNA polymerase, which reads the parental nucleotide sequence in 3' 5' direction and synthesize the new DNA strand in 5' 3' direction. Therefore, the newly synthesized chains grow in opposite direction. The strand in 3' 5' direction is leading strand and that in 5' 3' direction is the lagging strand. The lagging strand is discontinuous, producing Okazaki fragments which are joined by ligase. As one strand is discontinuous, therefore the DNA replication is semi-conservative. Now coming on to transcription. The template strand. And the template strand. And the next is the coding strand. Initiation is done by the RNA polymerase, which combines with the Rho factor at the promoter region. Further, the RNA polymerase synthesizes the RNA on the DNA template in the 5' 3' direction. Termination occurs with the help of Rho factor. The hnRNA form undergoes post-transcription changes called capping, tailing, and splicing. The next process is translation. Translation, the major role is played by the tRNA. The amino acid is activated. tRNA, also known as adaptive molecule, plays a major role in translation. The mRNA attaches to the anticodon loop of the tRNA, synthesizing the amino acid. Thus, the amino acid polypeptide chain is formed. This was the overall process of central dogma. Now here comes the second lab scene.
Now I have found the central dogma. My work is done. So let me go to the Dr. Watson to give all the thesis to him so that he can save it on computer. Good morning, Dr. Watson. How are you?
Fine, how are you?
Dr. Watson, can you store this thesis on central dogma on your computer?
Yeah, sure.
The next day when Dr. Green arrived in the laboratory.
Dr. Watson,
Oh, hi.
it's time to go to lab.
Yeah.
So get on your coat.
Yeah, I will.
Opening of the lab.
Oh my God! What has happened to our lab?
A thesis.
Our experiments, they all have burned.
Had they made the use of bioinformatics and used the computers, this would not have happened.
Dr. Watson, I have given you the thesis to store it on your computer. Yes, let's go to your home and just look those PPTs back.
Yeah, sure. Yeah, import it. I have stored it.
Bioinformatics is often described as being in its infancy, but computers emerged as important tools in molecular biology during the early 1960s. In 1970, Pauline Hoffman coined the term bioinformatics. Margaret Oakley Lehawk, who had been hailed by David Lippman, Director of National Center for Biotechnology Information, as the mother and father of bioinformatics. In order to study how normal cellular activities are altered in different disease states, the biological data must be combined to form a comprehensive picture of these activities. Therefore, the field of bioinformatics has evolved such that the most pressing task now involves the analysis and interpretation of various types of data. This includes nucleotide and amino acid sequences, protein domains, and protein structures. The actual process of analyzing and interpreting data is referred to as computational biology. There are different views of origin of bioinformatics. The term bioinformatics is a relatively recent invention, not appearing in the literature until 1991, and then only in the context of emergence of electronic publishing. The National Center for Biotechnology Information is celebrating its 10th anniversary this year, having been written into existence by US Congressman Lord Pepper and President Ronald Reagan in 1988. So bioinformatics has in fact been in existence for more than 30 years and is now middle aged. Major categories of bioinformatics tools. There are both standard and customized products to meet the requirement of particular projects. There are data mining software that retrieve data from the genomic sequence databases and also visualization tools to analyze and retrieve information from proteomic databases. These can be classified as homology and similarity tools, protein function analysis tools, sequence analysis tools, and miscellaneous tools. Here is a brief description of a few of these. Every day, bioinformatics is done with sequence search programs like BLAST, sequence analysis programs like the EMBOSS, statement packages, structure prediction, programs like Theater or PhD, molecular imaging, modeling programs like ResMod and What If. Some examples are FASTA, EMBOSS, ResMod, OPS. Thank you.